Hello, everyone. Welcome to our presentation about Nylon 6,6 and Nylon 6 synthesis using Biodesign and Generative AI. I'm Ariman Singh,
I'm Daphne.
and we're both from the Amati lab. Could you put in full screen also? Okay. First of all, we will introduce our project and give some background information. Then we'll move on to the objectives of our project, then our methodology, and our research design. Finally, we'll explain what we have done so far and what we plan to do in the future.
So to give an introduction about nylon, nylon is a family of polymers that is very commonly used. They're known for their strength, elasticity, and resilience. Nylon was the world's first fully synthetic polymer. And the most common types that are used today are nylon 6,6 and nylon 6. They're named for the number of carbons in the repeating units as shown in this image. And they're prized because they're lightweight, but also durable at the same time.
So here we have two graphs showing the US nylon market predicted from 2020 to 2030 and the predicted global nylon market from 2019 to 2026 on the right. The global demand for nylon has been growing exponentially for the last few decades, as they were first used in stockings in 1930 and are now used in textiles, automobile parts, plastic, and many other things.
So what gives you the best
So more on these two main types of nylon that we'll be focusing on. Nylon 6,6 has two precursors. The first is adipic acid, also known as hexadioic acid, and the second is hexamethylene diamine, or HMDA. And these two are polymerized through a condensation reaction that produces water, as shown. And some properties relative to nylon 6 are that it has a higher melting point and higher acid resistance. As for nylon 6, there's only one precursor, epsilon caprolactam, or just caprolactam. It has this ring shape so to polymerize it, you would open the ring in order to add more to the chain. And both nylon 6,6 and nylon 6 are aliphatic polyamides. They're linked by amide bonds. And aliphatic means that there are no double or triple bonds. And nylon 6 tends to be more flexible than nylon 6,6. So the conventional production methods of nylon 6,6 and nylon 6 are not the most sustainable. They use petroleum-based pathways. They also use a lot of energy input because the reaction requires high temperatures and high pressure. And they also release harmful byproducts. Both of these pathways start from benzene, which is a petrochemical and a carcinogen, which means it can cause cancer. And for nylon 6,6, benzene is converted to cyclohexane, and then to a mixture of cyclohexanol and cyclohexanone, and that is converted to adipic acid with a metal catalyst. This can include copper, vanadium, zinc, and lead, and especially for lead, it can be toxic. And this pathway releases a lot of nitrous oxide, which is a very potent greenhouse gas. It's about 300 times as potent as carbon dioxide. And adipic acid production accounts for about 1/10 of all man-made nitrous oxide emissions, which is a reason why we want to focus on making these methods more sustainable. And for nylon 6, benzene is converted to cyclohexane as well, and to cyclohexanone and cyclohexanone oxime, and then to caprolactam, which is the precursor to nylon 6. And this process releases ammonium sulfate waste, which is also harmful to the environment.
So here are objectives for our project. So our objectives are to identify environmentally friendly routes to nylon 6,6 and nylon 6,6, or nylon 6,6 and nylon 6, and identify alternative methods for adipic acid, HMDA, and caprolactam production. We also want to optimize the process to minimize generation of waste and the impact this process has on the environment in general. Additionally, we want to scale up sustainable methods while it currently has a higher cost and a lower efficiency. We want to reduce that cost and achieve higher yield while enhancing the procedure with further genetic engineering of microbes.
And moving on to our methods. And so we haven't started lab work yet, which we will soon. But we wanted to give an overview of some of the key techniques that we came across during our literature review. So there have been many recent advancements in more sustainable methods of nylon production. And these include biological strategies, which focus on engineering microbes. And the most commonly used are Cerevisiae yeast and E. coli bacteria, but recently they've also been developments with using other species of bacteria. And chemically, electrocatalytic methods can also help to reduce the amount of hazardous waste that comes from nylon production. And we also saw that using feedstocks from food waste is a more sustainable option for nylon synthesis.
So here are existing methods for adipic acid. So we start from benzene, which we transform into cyclohexane, which is then oxidized to yield cyclohexanol and cyclohexanon. Cyclohexanol is dehydrated to cyclohexene, which undergoes debromination and a zinc-mediated reduction which eventually forms adipic acid, which is a vital component for nylon. And concurrently, a galactoric acid undergoes O-acetylation to produce AGA, presenting a renewable alternative to conventional polymers. And this approach emphasizes the use of starting materials and renewable sources in synthesizing crucial industrial and environmentally friendly materials. And this whole process provides a sustainable alternative to other polymers, but uses renewable feedstocks.
As for bio-based methods, as early as 1994, a pathway has been demonstrated in using E. coli, converting glucose into several intermediates, DHS, PCA, and catechol, and then to cis-muconic acid, which is a very important molecule, which is a precursor to adipic acid, and this molecule can be found in many of the biological pathways to adipic acid production. And then it's hydrogenated to adipic acid. But since then, and especially after 2015, there have been significant progress in modifying E. coli in many different ways. And now they're capable of using a lot of other source materials beyond just glucose. For example, we can use xylose from hemicellulose feedstocks taken from plants, and xylose can be converted to PEP through glycolysis, which is a very important part of cellular respiration, and or E4P through the pentose phosphate pathway, which is also part of normal metabolism. And then both of these molecules can then be converted to DHS and follow this pathway to adipic acid. And yeast is also a very a common organism for biological adipic acid synthesis, and we'll be focusing on one method that it was the first time they showed the full synthesis of adipic acid in a single strain. And so they genetically engineered these by making a plasmid with this gene found in this bacteria species. And the expression of this gene and the synthesis of this enzyme that was capable of reducing a certain type of bond, it allowed for the conversion of glucose to muconic acid and adipic acid in that pathway. And finally, as mentioned, research has now expanded to other species beyond E. coli and S. cerevisiae, and this includes P. putida. So a former challenge of a biological adipic acid synthesis was that catechol, which is an intermediate in the reaction or in the pathway, it can be toxic to the host organism, but they were able to engineer higher catechol tolerance that allowed this to be more successful.
So here are existing methods for HMDA. So our current approaches to synthesizing HMDA, which is an important chemical used in the production of nylon and other polymers, they all rely on traditional non-renewable feedstocks. There's a growing interest in using fructose-derived feedstocks, which are more sustainable as they originate from biomass. So, however, there's a noticeable gap in research focused on developing these sustainable methods for producing HMDA. And this shows that we need further exploration and development in this specific area so we can transition from using fossil-based resources to renewable, more environmentally friendly alternatives. So there are existing methods for caprolactam. So we start by integrating titanium silicate 1 zeolite into a bed reactor, which enhances the catalytic efficiency by maximizing reactant catalyst contact. This improves conversion rates in suspended state. Silicate 1 zeolite combined with the moving bed reactor enables continuous operation and efficient catalyst use. This reduces the downtime through improved mass transfer. Amorphous nickel catalysts in a magnetically stabilized bed reactor allow for precise catalyst positioning and optimizing reactions. And this setup is useful in electrosynthesis and facilitates the transformation of nitrous oxide and cyclohexanone into caprolactam. And this whole method also cuts down the waste production.
And biological methods for caprolactam synthesis, which again is the precursor to nylon 6. A lot of these methods also are also done with E. coli. And one method is to use fermentation-derived lysine, an amino acid, and it's converted to this molecule and then to 6-aminocaproic acid, which is the linear form of caprolactam, and then it's converted to caprolactam. Or recently it's also been shown being converted directly to caprolactam, which then goes on to nylon 6. Another molecule you can use is 2-oxoglutarate or alpha-ketoglutarate, which is an intermediate of the citric acid cycle. And that can also be converted to 6-aminocaproic acid and then to caprolactam. And another source of materials that's applicable for all of these three monomers that we're focusing on, adipic acid, HMDA, and caprolactam, is food waste, or more generally biomass. And typical biomass feedstock includes corn stover, which is like the leftover parts of corn after the harvest. Or you can take it from food waste, like bread, fruit, vegetables, rice, etc. And some key substances are glucose, HMF, and levulinic acid. And these three can all be can all be converted to adipic acid, HMDA, and caprolactam.
So here we have a research design and approach. So our research question is, can we engineer microbes to produce all precursors for nylon 6,6 and nylon 6 without the use of inorganic catalysts or polluting chemicals? We'll be focusing on HMDA because of the lack of research that has been done on it compared to something like adipic acid. We'll be focusing on HMDA for nylon 6,6 and caprolactam for nylon 6. Our null hypothesis is biosynthesis in microbes is not possible, and our alternate hypothesis is some level of microbial synthesis of HMDA is possible. And so this is a design of our experiment. So to advance green biotechnology, we aim to replicate the existing bio-based methods for synthesizing adipic acid and caprolactam. This is very important in nylon production using renewable resources. Additionally, we plan to develop a biosynthesis route for adiponitrile precursor for HMDA, which we're going to use via engineered microorganisms or enzymatic pathways. Our focus includes bioengineering enzymes for hydrogen-driven biocatalysts to reduce environmental impact. So we will enhance bench-scale bioproduction efficiency through process improvements, emphasizing green chemistry principles such as reducing steps, minimizing waste, and optimizing the resource utilization. And our overall goal is to revolutionize the nylon production process, which we'll do by prioritizing sustainability and eco-friendliness. So currently our current status is we've completed the literature review and we're currently working on our review paper. And in the future, we'll be doing synthesis of precursors, our pathway design for HMDA, protein engineering. We'll be using AI for image generation of the products and synthesizing the products with 3D bioprinters.
And what we hope to achieve is optimization of sustainable routes to adipic acid, HMDA, and caprolactam production. And this includes using renewable carbon sources, reducing waste, making the process faster with less steps, maximizing yield, and improving the quality of the ventral nylon product. And this might include potential discovery of new microbial strains or new ways to engineer, of genetically engineering them for enhanced production techniques. Here are sources.
We would like to thank our advisor, Mr. Amadi, for all the help he has given us throughout the project. Thank you, everyone. Do you guys have any questions?